So I'm going to talk about the, protein adduct actually formation during electrophilic stress. See, as the question is good or bad, okay? So anyway, this slide shows the, a variety of post-translational modification of cellular proteins through, such as phosphorylation, acetylation, oxidation, and so on. Okay, so these are so important components, especially for cell signaling. And so post-translational modification can occur on the rhythm amino acid side chain, such as carboxyl group, hydroxy group, and the amino group, and the thiol. Okay, and it is already reported that there are 240,000 cysteine residue encoded in the human genome. Among them, about the 80 to 90% of such a, cysteine residue exists as the protonated thiol, SH, and disulfide bond, SS, and the ligand for zinc, and another, 10 to 20% of cysteine residue exists as deprotonated form, S- thiolate ion. So, such a protein thiolate ion easily, undergo oxidation, for example, by hydrogen peroxide, forming, sulfonic acid, SOH, or nitrosylation by nitric oxide, which is an inert, oxidant, okay, forming SNO. So these are, modified proteins, excuse me, is back to reduced to glutathione, which is an abundant nucleophile, antioxidant. So now you can understand that, so redox reaction consisting of the reduction of oxidation in protein are associated with, cellular signaling pathway, especially through modification of protein cysteine residue, okay? And we are, exposed to various xenobiotic electrophiles on a daily basis through, for example, the life environment and lifestyle and dietary habit, okay? So electrophile is highly reactive due to existence of, the electron deficiency moiety here in the molecule, so easily react with a molecule with electron rich, such as a protein cysteine residue, and forming a relatively, stable adduct. So we therefore saw the, so these electrophiles, just exhibits a different structure, right? But have the same chemical characteristic, just electrophile. So maybe such a, we saw the such a protein adduct is, involved in a structure change in protein and the modification of protein function, okay? So in 2002, John Florence, who was a professor of UCLA School of Public Health and the director for Southern California Particle Center, supported by Environmental Protection Agency, EPA, in the US, invited me, and finally I was selected as a sole grant member from Japan. And we found the 1,2-naphthoquinone. This is a structure here. Is contaminated in the PM2.5 and the vapor phase fraction of the atmosphere in California. And with naphthoquinones and other polycyclic aromatic hydrocarbon that have already identified from the atmosphere, and we found these naphthoquinones only exhibit, concentration-dependent, contraction of a guinea pig trachea. So we also found that 1,2-naphthoquinone, causes a contraction of trachea ring of guinea pig is attributable to, activation of PLA2, LO, and VR1, that's a vanilloid receptor signaling, leading to increase in calcium level, okay? And we also found the 1,2-naphthoquinone, phosphorylated, activated protein tyrosine kinase, and causes a contraction, but is blocked by genistein, which is a specific inhibitor for protein tyrosine kinase. And of interest, 1,2-naphthoquinone activated epidermal growth factor receptor, EGFR, and causes contraction, but is blocked by PD-153-035, which is a specific inhibitor of EGFR, suggesting, such a pharmacology action may be due to a covalent modification because, 1,2-naphthoquinone with electrophilic property just activated EGFR here, okay? But naphthalene, as indicated by N, without electrophilicity, nothing happened, so nothing pharmacological action, okay? So exposure of human epithelial A431 cell to 1,2-NQ just caused, activation of EGFR. So listen, coupled to inhibition of cellular protein tyrosine phosphatase activity in a concentration-dependent manner. So LA1, which is EGFR neutralizing antibody, just completely block the EGFR activation mediated by EGF, but just slightly inhibited EGFR activation mediated by 1,2-NQ, suggesting that 1,2-NQ effect is not due to a ligand dependent, okay? And also, Nick Tonks already demonstrated that EGFR, is negatively regulated by protein tyrosine phosphatase 1B with, thiolate ion due to very low PK value, okay? So, we look at the participation of PTP1B. So as you can see, the overexpression of PTP1B in the cell almost completely block the such EGFR activation mediated by, caused by 1,2-NQ, suggesting that the PTP1B is a main role in the, such a regulation of EGFR in the cell. Actually, we found the 1,2-NQ covalently modify the cellular PTP1B here as determined by a specific antibody against the 1,2-NQ we prepared, okay? So we also look at the in vivo study with the recombinant PTP1B. As you can see, the incubation of 1,2-NQ with PTP1B causes time and concentration dependent inhibition, reduction of its catalytic activity decoupled to a covalent modification of PTP1B protein. So again, 1,2-naftoquinone with electrophilic property just modifies PTP1B and inhibited its catalytic activity. However, either trans 1,2-dihydroxy 1,2-dihydronaphthalene as indicated by DDN or naphthalene indicated by N. Never covalent modification and thereby never, inhibition of PTP1B activity. Further supported the 1,2-NQ actually modifies PTP1B and causes EGFR activation. So as expected, the MADI-TOF mass analysis reveals that the covalent modification of PTP1B by 1,2-NQ is indeed system 121. So our data shows that, so 1,2-NQ covalently modified the PTP1B, so leading to a reduction of its catalytic activity, okay, because with the cyanide ion, thereby activating EGFR, okay? So, beside the PTP1B EGFR signaling, so it is already reported that there are other redox signaling pathway consisting of sensor protein with cyanide ion and effector molecules such as kinases and transcription factor. So under basal condition, so effector molecule is negatively regulated by sensor protein. So I just show the very representative redox signaling pathway, okay? So, for example, PTP1B EGFR is linked to cellular proliferation. KEAP1 NRF2, that's a very popular yesterday, huh? Also, Songgong. Yeah. And is oxidative stress response, and HSP90 and HSF1 is linked to quality control of cellular protein, and P10 AKT is also cell survival. So under oxidative stress or inflammation condition, so these effector molecule as indicated by pink color is activated after the decreased activity of each sensor protein, okay? Due to oxidative modification, we therefore thought that, maybe sensor protein are molecular target for environmental electrophile through covalent modification. This is a oxidative modification. This is a covalent modification, okay? So today I'm gonna just introduce you KEAP1 NRF2 pathway because very popular, okay? So transcription factor NRF2 negatively regulated KEAP1 which interacted with CAL3. So under basal condition, the NRF2 is readily undergo degradation by ubiquitin proteasome system. So leading to a very, very minimal level of NRF2 in cytoplasm. So we also exposed to a variety of electrophile and found that these electrophile all modified the KEAP1 through cysteine residue, sometimes overlapped, sometimes so different. Okay? As a result, so NRF2 newly biosynthesized just go to nucleus because transcription factor, right? and meet partner protein small MAF and forming heterodimer, such as NAF2 and small MAF, thereby activating, upregulating the downstream genes such as antioxidant protein and phase 2 xenobiotic enzyme and phase 3 transporter, okay? And methyl mercury is a causative agent of Minamata disease, which is, it's kind of a tragedy happened in Japan, okay? Just affecting central nervous system. And an issue we should pay attention is that methyl mercury accumulated in the body through the food chain and the bioaccumulation, so leading to in the ingestion of a large edible fish such as a tuna, okay? You still take methyl mercury if you like Japanese food. And methyl mercury modifies the protein through the cysteine residue because an electrophile. And, but methyl mercury is detoxified by GSH. So I described here, now you can understand the, these, protein as indicated by blue color, so associated with detoxification of methyl mercury and the exclusion of into extra space is cooperatively regulated by a transcription factor NRF2. So we saw the maybe methyl mercury modified KEAP1, okay? So as expected, so methyl mercury modifies KEAP1 here. And after exposure, now you can see, methyl mercury exposure just causes activation of NRF2 in a concentration-dependent manner and upregulated GCL isoform and GST isoform. So to look at the role of NRF2, we used with knockout mouse for NAF2 and KEAP1, okay? So NAF2 deletion, the cause is, significant increase in mercury content in the cell, okay? Thereby, enhancing methyl mercury concentration dependent cytotoxicity. By contrast, NAF KEAP1 deletion to causes a permanent, activation of NRF2, just markedly increase MRP1 isoform, so leading to, decrease mercury level in the cell, thereby, blocking cytotoxicity mediated by methyl mercury, suggesting, NAF2 is a key molecule for repression of methyl mercury. We also look at the in vivo. So this is, hindering flaccidity, which is a very typical behavior for methyl mercury intoxication. So under this condition, so nothing happened of hindering flaccidity, and mortality was seen in wild type mice. However, NAF2 deletion causes obvious, hindering flaccidity and time-dependent mortality. So further supporting NRF2 is pretty good for repression of electrophiles such as methyl mercury, okay? So as I already told you, P10 AKT signaling is associated with cell survival, right? But listen, exposure of this cell to methyl mercury, facilitated nuclear translocation of AKT as determined by not only Western blot, like also staining study, and we also found the methyl mercury activated P10 and its downstream proteins, which is it's kind of a transcription factor, CREB, and finally upregulated antioxidant protein BCL2 in a time-dependent and concentration-dependent manner. Okay? So, most people just believe that the methyl mercury is very toxic, very bad, right? So, to understand the biochemical meaning of activation of P10 AKT signaling mediated by 1,2-NQ, we pre-treated with wortomannin, which is a specific inhibitor of PI3K kinase AKT signaling. So now you can see methyl mercury causes apoptotic cell death, but wortomannin enhances such a cytotoxicity. Also, we found the wortomannin pretreatment enhance methyl mercury mediated cytotoxicity. Again, the knockdown of BCL2 also enhance methyl mercury, so suggesting the activation of AKT CREB BCL signaling mediated by methyl mercury, but listen, at low dose is involved in an adaptive response against methyl mercury. However, increasing concentration of methyl mercury completely block activation of PTP and also CREB block activation, so downregulation of BCL2. Such a phenomena, specific, those specific alteration in signaling is also confirmed by luciferase activity for CRE. Okay? So this slide shows the cooperative modification of the P10, AKT, and the CREB. Okay? In this assay, we just established its kind of assay. So in this assay, decrease Western blot intensity is associated with increase the covalent modification methyl mercury. Now you can see the, P10 is very sensitive to methyl mercury, and increasing concentration of methyl mercury is also CREB. So methyl mercury modifies the CREB as well, but AKT is almost nothing happen. Okay? And in a separate experiment, we reported that another environmental electrophiles such as 1,2-NQ just modifies the system 286 in CREB. Okay, so resulting in interference between CREB and CRE. Okay, thereby downregulating the BCL2, so causes apoptotic cell death. So according to such phenomenon, now you can see methyl mercury also modifies system 286. Okay, through because this modification is declined by mutation of the system 2 setting. Okay, so just a brief summarize here. So basal condition, AKT is negatively regulated P10, but at low dose, methyl mercury just selectively modifies P10, okay, and leading to its catalytic reduction of catalytic activity, thereby the activation of AKT and CREB, and finally upregulating BCL2 involved in cell survival. But, at high dose, that's terrible because, methyl mercury is electrophile. So easily modify the, extensively modify the cellular protein, including CREB, through system 286, thereby blocking such interaction between CREB and CRE. So finally causes apoptotic cell death. Okay? So today, I don't have enough time. So let me show the, this is an overview we got. So, through, life environment such as atmosphere and tobacco smoke, and through dietary habits such as coriander and rice on the large edible fish such as tuna, So these electrophiles just attack to, redox signaling pathway. So finally, activation of effector molecule coupled to inhibition of sensor protein. Just I summarize our publication of original article and review article. So finally, I just cited the cartoon from the Casaret and Dole Toxicology and the 9th edition. Maybe top page or something like that. So now, really understand, the such a small amount of falling material have little effect. Nothing happen on the human body, but large amount do. Okay, so, but very nice cartoon, but there is no mechanistic detail. So today I'm gonna insert it. Okay, especially in case of electrophilic stress. Oh, it's fine because we have the adaptive response system. And today I don't have enough time, so we have the protection against sulfur adduct, okay? But too much, terrible, right? Doesn't make sense, So this is acknowledgement slide. So let me introduce just briefly. In Japan, it's customary to wear a red vest with the name of Chanchanko when celebrating 60th birthday, okay? But actually, I hate that, because, anyway, I don't like. So my graduate student gave me a red color, but down vest made by Canada Goose, okay? Now the, I met, Motohiro Nishida over there, and we started the collaboration study from maybe 2008 or 2009, huh? And anyway, thank you very much, Moto. Thank you for, because he gave me a special position, especially appointed professor in Kyushu University. And we also, I wish to thank for so many Japanese grants and the US grant as well to support our research nicely, okay? So this is a final slide, because a special thanks to Professor Sang-Gon Kim and Yeonjun Sir. Yeah, unfortunately, he already left back to Seoul. And because, he joined the annual meeting of JSOT, Japanese Society of Toxicology in Fukuoka, okay? And also invited me here. 감사합니다. That's all.